I'm Des Higgins. I'm Emeritus Professor of Bioinformatics in the School of Medicine in University College Dublin, Ireland. And I just retired last September after about almost 40 years of working as a bioinformaticist. I started as a bioinformaticist before the word was even in common use and before people realized how important bioinformatics would become. So I started in 1985 in Dublin as a postdoc. And I came to EMBL in 1990, initially as a postdoc and then as a staff scientist. And I stayed at EMBL until the end of 1996, having moved to the EBI in 1994. So I was at the EBI for 2.5 years.
Great. So it seems like you have been witnessing, again, the beginning of the bioinformatics at the EMBL, and perhaps also the beginning of the EBI as an institute.
Absolutely.
Or at least the establishment and setting up of that for part. It was probably already set up when you came.
No. So I can explain that. Yeah. So bioinformatics in 1985 was something that some people got very excited by. A lot of biologists who liked writing computer programs but didn't have much to write computer programs about suddenly realized that you couldn't look at DNA sequences. You couldn't even look at them without software. Because once DNA sequences went more than 5 or 6 lines long, it was difficult to just look at them by eye. And so people working on phylogenetics, structural biology, people cloning and sequencing genes, just genes of interest, all had to start getting interested in bioinformatics. But the first bioinformatics meeting I went to in 1986 was in Waterville Valley in New Hampshire, and there were 200 people at it. And at that stage, the entire population of bioinformaticists on the planet was about 400 people. So half of the bioinformaticists in the world were at this meeting. And that was 1986. But it just felt exciting. It was a Wild West spirit about bioinformatics. We felt we were the chosen people, that in a while, bioinformatics is going to be really important for everything in biology. And this turned out to be even more true than I expected. I can remember Michael Ashburner. Michael Ashburner was one of the people responsible for persuading EMBL to set up a nucleotide sequence database. And that was in 1981, and that went on to become the data library, which in 1994 became the EBI, the European Bioinformatics Institute. And I can remember hearing Michael Ashburner in 1990 giving a talk in Dublin to a genetics society, and he was asked what he thought the most exciting thing in biology was. Now, there are lots of exciting things in 1990. A lot of molecular biology was very exciting. People were discovering amazing things in many areas. But Michael said he reckoned bioinformatics was the most exciting area, or the most exciting growth area. And he was a very visionary man. I personally knew Michael Ashburn. He wrote me one of the references for my current position in Dublin. But he also, I can remember him sitting at his computer in Cambridge, typing in keywords, biology keywords like kinase or ribosome or ribosomal protein and so on. And he started by trying to make a collection of all of the possible biological keywords for Drosophila. And that ended up becoming GO, the gene ontology system.
Okay.
Which revolutionized a lot of gene searching and a lot of microarray analysis about 15, 20 years ago. So Michael is a very influential man.
Yeah, well, that's very interesting because the approach is, I guess, the classification approach, right? You want to give label to that so that labels will be a reference and then...
It's labels, exactly. That when you have DNA sequences, one of the problems is if you have a big DNA sequence, it's just pages of letters, and you need labels. And so you can try to create labels by analyzing a sequence with computer software, looking for features, looking for homology, looking for similarity with other sequences. And having done that, having identified maybe where the functional RNAs are, or the messenger RNAs, if there's a gene product, you would like to know what that gene product is, or to be able to put a label on it, so that that can then go in its box on a shelf with all of the other genes in the genome.
And I think this is key to build a nomenclature that a computer can actually work with.
Exactly. So it makes things searchable. It means if you're looking for a kinase, you can look for kinases. Without keywords, the only other way to look for a kinase is to look for similarity against a known kinase using similarity search software. So that's sort of classic bioinformatics. That's the kind of stuff that I was interested in.
Yeah, so it's nice because you go from ordering stuff in a, let's say, physical world to ordering stuff into a sort of an electronic world. So it's very interesting that one of his occupations or preoccupations was typing in labels that could be useful later on. But speaking of things that could be, let's say, where you had to make order even before filling up those categories, I mean, could you envisage back then that you would be flooded with so many sequences? I guess, sequences was quite hard back then.
So back in the early days, I can remember the very first DNA sequence database I used, that I had access to. It came from here, across the road in the older EMBL building, on half-inch tapes. So they looked like big reels looked like reels of audio tape. You can imagine from an old music studio. And these were posted from EMBL Heidelberg once every 3 months. And they would arrive in a big parcel in your lab in Dublin. In my case, you'd open up the parcel, load the computer tapes, and the first edition of the EMBL database that I saw had 4.5 million nucleotides of DNA. So that was 1985. So DNA sequencing had only been invented, if that's the right word, in the most widely used method by Fred Sanger in Cambridge in 1977. So within 8 years, there were 4.5 million nucleotides of DNA and about 5,000 to 10,000 genes, or 5,000 to 10,000 pieces of DNA, some of which were
which was already genes. Quite a good
So it was already big, but in those days, it was small enough where people could print the whole thing out if you really wanted it. You could buy it as a book. Or when I say buy it, I think it was free. EMBL gave them away free. And initially there was one book, and it was called the EMBL Nucleotide Sequence Database. It would have a date on it and a release number. And you could look up in an index and you could flick through the whole thing.
Oh, this is fascinating. The EMBL had the book of life at some point.
Well, it was a book of a tiny little piece of life. So if you're to do the equivalent for one human genome, then somewhere in the Wellcome Trust have shelves of books with the genome printed out. But already this was almost too big to be done on paper. And within a few releases, that was abandoned because it simply wasn't practical anymore.
Yeah, it's like a telephone index that gets...
It was going like that, yes. So when I went to this bioinformatics meeting in 1996, I met half of the most famous bioinformaticists on the planet. So I met Lipman and Pearson, Smith and Waterman. I met Wally Gilbert, who wasn't really a bioinformaticist. I met all kinds of famous people. And I met Graham Cameron, who at this stage was head of what was then known as the data library. So the data library collated the sequences, typed them in, and released them as releases of the EMBL nucleotide sequence database. And I got talking to Graham, and that was 1996. And a few years later, the data library was expanding because there was so much work to be done. And the data library advertised for some software engineers, and I applied for one of the jobs, which I didn't get, but that led to a process whereby I applied for funding and came to work for Graham in the data library on the 3rd floor of the old building over the up run. And I arrived then in EMBL in 1990. And then Graham was very busy after a few years setting up the EBI. And when the the EBI was initially set up, it basically was the data library from here, which simply moved, and I moved with it. So Graham was a very wise and very wonderful mentor. So I worked for him from 1990 to almost 1997. And I've great memories of his wisdom and his wit and his leadership.
You know, we know from history that, you know, you have been developing an algorithm for comparing sequences. And so, and this is actually even before your time at EMBL, right?
Yes.
And so it would be great to have a sense of how you went about that. So what was the genesis of that? What were the challenges, and was there any aha moment that led to actually getting something that would work?
So the way I got into multiple sequence alignment and then developed the first Clustal, it started in Dublin in the late 1980s, and I needed to write software for carrying out various sequence comparisons, because people would come to me with a DNA sequence that they'd spent maybe 3 years cloning and sequencing and wondered what it was. Was it a gene, or was it a protein, or what was it? And it would come to me with a gene on a piece of paper, and I'd take the piece of paper to a terminal on a mainframe computer and type in the DNA, and then analyze it, look for open reading frames, and so on. But the research in our lab, at that stage I was a postdoc for a man called Paul Sharp, who is currently Professor of Genetics in the University of Edinburgh. I was his first postdoc, and he was my first postdoc boss. And his lab worked on molecular evolution. And in order to make a molecular phylogeny, you need to get a collection of sequences and line them up over each other so that you can see homologous residues in columns. And we did those by hand. So initially this was done with pieces of paper and colored pens, sometimes a scissors. You would create a gap by using a scissors.
Wow.
Later we used word processors. And so it was an incredibly tedious process. It was very unsatisfactory because it was, you can go a bit blind. If you look at a big collection of sequences, you can go a bit crazy trying to figure out how to line them up properly.
Exactly.
And I thought there has to be a way of doing this automatically. If we're doing it by hand, there must be an automatic way. And it turned out that there were some methods for doing it, but this was before the World Wide Web. And so software and information about software spread very slowly in those days, sometimes by word of mouth. And to get a piece of software, you often had to write a letter to someone who would send you back one of these half-inch tapes, or maybe a floppy disk if you were lucky. And I couldn't get any automated software for doing multiple alignment. And there were some methods published that described how to do it. The earliest was from a man called David Sankoff from 1973 or 1974, which is before DNA sequencing. And these were 5S ribosomal RNAs, because there were methods for sequencing small RNAs before Sanger dideoxy sequencing. And I adapted some of these methods, and I also made them work on a PC. So in those days, the PCs we're using were what were called IBM PCs, although they weren't necessarily made by IBM, and they had 640K of memory. So that's half of 1 megabyte, which is 1,000th of a gigabyte. So it's hard to imagine how tiny this amount of memory is. The first machine I used had an 8-bit processor. These days machines have 64-bit processors. So these were incredibly old, weak, tiny computers, but I figured out how to make multiple alignment work on a PC. And initially I did this for me, because I wanted multiple alignments, and so that I could stop doing these by hand with scissors and colored pens. And the lab were very pleased with this, and we wrote a paper. And unfortunately, in those days, the only way of distributing the software was where we told people to send us floppy disks in the post. So these were about this size, 5 1/4 inch floppy disks. They were floppy and they were 5 1/4 inches.
Yeah, I remember those. I
you
got
do
to
remember?
work with a bit of those. Yeah, they were very delicate. Exactly because
you
they
look
were
too
floppy.
young, and they were very delicate. So people would send us floppy disks in the post. And in the first couple of years, by the time I came to EMBL, so I was still working on Clustal, I came to EMBL with Clustal as my ongoing project. And we'd managed to send out about 200 copies in the first 2 years until Roy Omond and Peter Sturr in the data library, and Roy Omond, who was group leader, head of the computer lab in EMBL, set up an email server where you could send a request by email, and if you put in the body of the email, get software : Clustal, Clustal would be mailed back to you, encoded in an email.
Just as a sort of
It was
run code.
in something like a zip file.
Okay.
It was an encoded file that could be transmitted over email.
And listen, I mean, do you think, you made me curious with this idea that you were physically cutting sequences in alignment. I mean, did that inform the way that you did it electronically, or you had to change completely system and start from scratch to think about comparisons and matrices and God knows what else?
Yeah. So as to whether as to whether we learned how to make better multiple alignments automatically by doing them by hand or not, I'm not sure. Mm-hmm. The fundamental way that you tend to do it by hand is block by block. You look for blocks. Mm-hmm. And in a simple case with a single domain protein, if it's proteins, the blocks could correspond, say, to alpha helices. Mm-hmm. And they might be, and beta strands, and they might be separated by gappy places, which would be the loops. Mm-hmm. But you tended to look for the blocks and then slide sequences in, try and build up the blocks. Whereas, and some multiple sequence alignment methods still do that, but computationally it's extremely difficult. It's and especially if you have repeats. Because imagine if you have repeats, even just 2 copies of a repeat, then if you're trying to line up members of this repeat with members of this repeat, they can confuse each other. You can end up with pieces in the wrong block. So you tend to do it the other way around. You do it by building up the alignment, starting with the most closely related sequences, the most similar sequences, and you line those together, and you then build the alignment out by adding sequences gradually, but you add entire sequences.
I see.
So, and that's not because necessarily you want to do it that way, but that's just, it's by far the most efficient way of doing it computationally. And it does make some biological sense in that, in principle, the sequences evolved by diverging.
From similarity and radiating.
Yes.
Yeah, certainly. And certainly, yeah, you were mentioning phylogeny and evolution, but we should remember that one of the key advantages of having a software like that is also assembling rather larger sequences, because I guess in the early days where you couldn't sequence like long bits, and still now you had to rely on comparison to look at overlap. Did your algorithm help also in assembling larger sequences and eventually contributing
so not so much in say genome assembly. Mm-hmm. So that's a different problem. That problem started in Fred Sanger's lab. Mm-hmm. Software, a bioinformaticist called Roger Staden wrote software for assembling this way. But even in the mid-1980s, there were some long sequences. HIV, the AIDS virus. The genome is about 10,000 bases long. And there were some complete genomes of HIV, which needed to be lined up, although usually for phylogeny. Phylogeny for HIV was very important in figuring out where the virus came from. They tended to use pieces of the Pol protein, pieces of the polymerase. And also, there was a limit to how long the sequences could be. If you double the sequence length, then some of the internal parts of Clustal, the time and memory go up at the square of the sequence length. So if you double the sequence length, it takes 4 times longer.
Okay.
For some of the internal steps.
So when you get to a certain size, it becomes very difficult.
It became difficult. So back in those days, the very first Clustal we released could align 1,200 residues, DNA or RNA or protein. These days, the biggest ones we've ever done were we aligned some titin proteins, and so they're almost 30,000 amino acids long. But we haven't been able to do that until fairly recently. Fortunately, there are almost no proteins even remotely as big as titin.
Yeah, I guess it's one of the biggest proteins that genomes can encode.
I think it was the biggest protein, and the first person to sequence titin, the first sequencing of titin happened here at EMBL.
Okay. This is a time where you were at the EMBL, or?
Yes, and so one of the wonderful things about EMBL was you would meet people at coffee and at beer and at parties. EMBL was always having EMBL-wide parties or just dinner in people's houses, going to seminars, and you would hear people ask questions and you think, oh, they must know something about something I'm interested in. And you just go and talk to them. And so I worked on the evolution of titin for a while because Siegfried Labeit and Matthias Gautel and Toby Gibson, my long-term Clustal collaborator, he was already interested in titin. And they were sequencing, initially they sequenced about a third of titin here. They ended up sequencing the whole thing after I left EMBL. So that was one of my collaborations at EMBL. And these collaborations just started out of nothing. You'd be having a coffee with someone and someone would say, hey, you can make trees. Will you please help me analyze my sequences?
Yeah, absolutely.
Yeah. That's how I met Toby. That's how the most famous thing that happened to Clustal was ClustalW and ClustalX. And they came from a collaboration with Toby Gibson and Julie Thompson, who were in a different lab in EMBL. And I knew Toby was making multiple alignments by hand, and I with colored pens, and I said, I've got a program that can do that. He said, your program won't be as good as what I can do. And so we collaborated, and that ended up becoming ClustalW and ClustalX. And so the environment for collaboration at the time was really first class. And a lot of people had the freedom to be able to put someone on a new project immediately. Like you could be having a conversation with someone over beer the night before, and next morning you could have people working on the new idea.
So that kind of flexibility.
The flexibility was outstanding, and the atmosphere was very open.
Yeah,
people wanted papers, they wanted collaborations, and they knew one way of doing that was to collaborate like crazy.
But I guess we were also witnessing, coming to the end of the 90s, more and more sequencing, and I guess those were the years in which we had, started to have the methods to do sequence entire genomes, and we start to get into races for the C. elegans genome, the fly genome, the human genome, and there were also issues with people thinking that these sequences could be profitable and patentable. And so what is your view about these things, and can you tell us a bit of a story or how you saw these from your eyes as you were doing these at the EMBL and after the EMBL?
So from the beginning, the GenBank and EMBL nucleotide sequence databases and DDBJ in Japan, there was a three-way partnership around the world. And from the beginning, everything in those databases were freely available. You didn't have to pay. Individual sequences might have been published by people, So you would have to acknowledge those people if you analyze those sequences and made use of them. But people understood that if you put a sequence into a database, that it was publicly available. And this was enforced. EMBL and GenBank and DDBJ went to great lengths in the late 1980s and early 1990s to make it compulsory that if someone published an analysis of a DNA sequence, someone might have cloned a gene, and they would write this up as a paper, and they might say why that gene is interesting. But if they wanted to do that, they had to make the sequence available as a condition of publication. Most journals dragged their feet over that because that's an extra red tape step. Nucleic acid research led the race, or it wasn't a race, led the field in making it mandatory, so that you could not publish an analysis of a sequence without making the sequence available, and making it available meant it had to be deposited in the databases, in one of the databases. The databases shared information. So from the beginning, there was a feeling that DNA sequences belong to the world and belong to the people who funded the research that paid for the sequencing.
Taxpayers, mostly.
Taxpayers. And if companies wanted to sequence privately, which some did, that was fine. But you couldn't then publish analyses of those sequences in any of the normal journals. You could keep the sequences in-house and exploit them and sell them. You're perfectly entitled to do that, and some companies did. But once the sequences went into the databases, they were freely available. And this had to be fought for really hard. A lot of people didn't see the point of this and didn't think it would matter. It came to a head when the human genome when the plans were underway to actually sequence the human genome. As a trial run, C. elegans and Drosophila were sequenced. And there was a move at one stage to keep Drosophila private. Oh. But Michael Ashburner and various other people fought to make sure that it would be freely available, and it was, even though the sequencing was done by a private company, by Celera Genomics. And so then when the human genome was sequenced in a public consortium led by the Sanger Center and various other places, by John Sulston and others, and by Celera Genomics, both sequences were made available. They were made public.
Yeah, so that was, I remember, because I was already in research back then. We're talking about the beginning of the 2000s. That I think the tradition of openness was really what really led, I guess, the American government and more globally to really convince Celera that they couldn't really profit out of that, and they would better play nice.
And David Lippman, who at that stage was head of the GenBank project in the National Library of Medicine in the NIH, he championed this vigorously. David Lippman was also the author of one of the most widely used pieces of bioinformatics software ever written, BLAST. Mm-hmm. And from the beginning, from day one, even before BLAST was published, they were giving the code away.
So another question is, now in thinking about after the EMBL and your career building up to this time and witnessing the explosion of DNA or RNA or nucleic acids derived data mostly, what do you think of where the field is going? Because I think we're flooded with a lot of data, and certainly one of the challenges is trying to find automatic or artificial intelligence way to make sense out of this data, and it looks like there is a big bottleneck there in bridging what we get in terms of observation to real understanding.
So as to where the field of bioinformatics or field of genomics, let's say, is going, before the recent developments in AI, I would have said I haven't a clue. I've never been very good at predicting anything in genomics or in bioinformatics. I've simply gone with the flow. And now the volumes of data that can be produced and the types of data is simply astonishing. And as to what you do with all that, I mean, there are very nice applications, right, such as eDNA. So I started off life as an ecologist and a taxonomist. And so if you want to find out what creatures are living in a river, you just take a water sample and sequence
environmental DNA.
Environmental DNA.
Yeah.
So there's some fantastic applications. But as to on the research front, where bioinformatics could get exciting, the recent developments with what's it called? ChatGTP?
Yes. Yeah, we've been discussing that a lot among alumni.
It's frightening and incredible and interesting, and the power is phenomenal. So-called AI methods have cracked protein folding. Yeah. And that's astonishing. A lot of people said that would never be solvable. I can remember Graham Cameron making the observation that about artificial intelligence, that back in the 1970s and 80s, people tried to write software to play chess. And they tried to make the software intelligent. And so people tried to make the software learn chess skills, strategy. And that turned out to be very difficult to do. In the end, the winning methods for chess turned out to be brute force and libraries. So the best chess programs stored away libraries of all possible openings and some number of moves after that, not many, up to a certain depth, and they would memorize those. So they already knew the optimal move for the first 3 or 4 moves. And after that, it was simply brute force. And so it's a combination of memorizing, of learning what's out there, by copying blindly and unintelligently, and some rules built in. And this seems to be how the protein folding problem has been cracked. The protein folding problem, it also makes use of multiple sequence alignments, very big ones, very simple multiple alignments sometimes. But the methods that are used to solve protein structure, I don't think they're very profound. I simply think they're well polished and they're able to learn from vast numbers of examples.
Okay. So you say simple operation, but at scale.
Using scale.
Yeah.
Using the scale.
Some sort of emerging properties or really a jump in the quality.
For example, with the scale, so people always said that if you had a multiple alignment, if you have multiple alignment of RNA sequences, you can look for pairs of bases that seem to be associated, because you get a signal. If you get a pair of bases, that base pair in a stem, in a double-stranded region of an RNA, then if you take 2 of those positions and get a multiple alignment of a set of those sequences of homologs, you can see that those positions share a certain amount of information. So if you change a base in one sequence, the matching base also has to change. And people try to do this with proteins, try to look for a shared signal in amino acids that were in contact. And this failed miserably. I knew of about 20 different attempts at doing this in the 1980s and 1990s, until there were enough sequences, until you had tens of thousands of sequences. And then it started working.
You know, the feeling of what you were discussing before, of people, including you, being adamant that you had to share your knowledge, I think it's a very gut feeling, right? You think you're doing something that's positive.
Yeah.
And culture is part of it. So let's talk about your feeling for openness.
Yeah. So the culture of openness in EMBL, it was remarkable. It felt like such an open building. There were seminars happening all the time. I remember if you got hard writing software, whatever, think, oh, there's a seminar on, I'll go, and it could be on something random. And people would tell everyone what they were working on. And people felt safe to do that. And I think in the long term, people benefited, the EMBL certainly benefited from this culture.
Well, thank you so much. It was a very interesting and enriching conversation.